student, welcome to the Medical Genetic course. Today's lesson is titled Genetic Mutation. By the end of this video, you are expected to define genetic variants, identify types of gene mutation and their effect on DNA and protein sequence, understand the functional consequences of mutation on the protein product. In this lecture, you will also define the meaning of genetic variants and the difference between mutation and polymorphism. You will also learn about the types of the mutation that affects the DNA sequence, which subsequently affect protein structure. You will also know the functional consequences of mutation on the protein product. What is genetic variant? These are changes in DNA coding or non-coding sequence. There are two types of variants. Normal variants with no obvious effect upon phenotype, it's termed polymorphism. It's called SNPs, which stand for single nucleotide polymorphism. These are changes in the sequence with no effect on phenotype. It occurs on average every 300 nucleotides. The majority of these are benign. On the other hand, a mutation is defined as heritable alteration or a change in the genetic material. Mutations drive evolution, but can also be pathogenic. In human genetics, the term mutation has often been reversed for DNA sequence change that cause genetic diseases and are consequently relatively rare, with a population frequency less than 1%. Mutations can arise through exposure to mutagenic agent, but the vast majority occur spontaneously through errors in DNA replication and repair. The first type is spontaneous mutation. For example, it arises through errors in DNA replication during cell cycle. These are internal events. The second one is induced mutation by a mutagen. For example, external agent that induce mutation such as radiation or chemicals. Where does the mutation occur? It can happen in germ cells or somatic cells. Somatic mutation may cause adult-onset disease such as cancer, but cannot be transmitted to offspring. However, a mutation in gamete can be transmitted to future generations unless it affects the fertility or survival into adulthood. We will focus on mutation that happen on level of single gene. Mutations are divided into two groups according to their effect on polypeptide sequence of the protein. The first one is synonymous, which is silent mutation, when a single base substitution in a codon results in the same amino acid because of the degeneracy of the genetic code. This is called silent mutation because it does not alter the function, structure, and properties of the protein. The second type is non-synonymous. When the mutation leads to the change in the protein sequence, it results in the alteration of the function and properties of the protein, which could lead to diseases. So, what are the types of gene mutation and their effect on DNA and protein sequence? Point mutation or base pair substitution means a change in a single DNA base. It consists of missense, nonsense, or silent mutation. The second type is the frame shift, which is the result of insertion or deletion of one or more base pair. These mutations, which can result in extra or missing amino acid in a protein, are often determinantial. We will talk about it with more details in the next few slides. The first type of point mutation is the silent mutation, which does not change the protein sequence because of degeneracy of genetic code. It also has no effect on the protein sequence or function. That's why it's called silent mutation. In the figure above, the point mutation changed the one nucleotide in the DNA sequence from adenine to thymine. However, the genetic code GTA or GTT is representing the same protein, which is valine. The second type of point mutation is the missense mutation, which occurs when one base pair in the DNA sequence is substituted or changed, as you see in the DNA sequence, in which C is substituted with an A, which result in changing of the amino acid to another different one. An example of the point mutation that cause sickle cell disease is a missense mutation. The DNA sequence CTC encodes the mRNA codon CAG, which specify glutamic acid. However, in sickle cell disease, the mutation changed the DNA sequence to CAC, which encodes GUG in the mRNA, which then specify valine. This mutation changes the protein shape, which alters its function and causes sickle cell anemia. The third type of point mutation is nonsense mutation, which is the change of single base pair which produce one of the three stop codons, including UAA, UAG, or UGA in the messenger RNA, which results in premature termination of translation, resulting in truncated protein, which can greatly influence the phenotype. Therefore, nonsense mutation result in changing the amino acid sequence to stop codon that result in termination of translation, which results in huge effect on the protein than the missense mutation. The second type of mutation is frameshift. Insertion is a type of a frameshift mutation. If a mutation involves the insertion of nucleotides that are not multiple of 3, it causes shift in the translation reading frame. Frameshift mutation have more significant effects on the protein than the point mutation. Therefore, the codon sequence will be changed, and this could result in completely different sequence of amino acid from the point of insertion or deletion on forward, as we see in the picture. If an insertion occurs, it results in alteration of all codon downstream from the site of insertion. If there is insertion of 1 or 2 base pair or more, not a multiple of 3, it disrupts the reading frame. Also, in frameshift mutation, if there is a deletion in 1 or 2 base pairs, it results in alteration of all codons downstream from the site of deletion. Loss of 1 or 2 base pair or more, not a multiple of 3, is harmful and results in disruption of reading frame, and it has drastic effect. Now we will talk about the functional effect of mutation on the protein level. It's useful to think of mutation in terms of their effect on the protein product. Broadly speaking, mutation can produce either a gain of function, a loss of function, or dominant negative effect. Gain of function, as we see in the picture, results in novel protein product, or it can also result in overexpression or inappropriate expression in the wrong tissue or wrong stage during development. Loss of function mutation result in reduce or eliminate protein product. Loss of function mutation are often seen in recessive diseases, where the mutation result in the loss of 50% of the protein product, such as metabolic enzymes. But the 50% that remains is sufficient for normal function. The heterozygous is thus unaffected, but the homozygous having little or no protein product is affected. Lastly, the dominant negative mutation. It results in a protein product that not only is abnormal, but also inhibit the function of the protein product by the normal allele in the heterozygous. Lastly, these are the references used in the lecture, and you can always go back and read. In summary, we have talked about genetic variants, which include polymorphism and mutation. the types of gene mutation and their effect on DNA and protein sequence. We also talked about the functional consequences of mutation on the protein products.